And this is a presentation by Soil and Water Conservation Society of Metro Halifax, conducting its regular monitoring of lakes. It started actually in the early 80s and more intensely since 1990. This particular project is on macroinvertebrates, that is those life which lives on the bottom or in the bottom of lakes, and they're usually the infantile or the beginning stages of insects, and all stages of mollusks and others. And my name is Shalom Mandeville. I'm the coordinator and founder of this group. And hopefully all of you will enjoy watching this to see how people can make a difference. And our society's theme is Think Globally, Act Locally. From now on, most of the conversations here you will hear are from two up-and-coming biologists by the name of Monica Gertner and Angela Jarvis. What they're basically doing are collecting these macroinvertebrates through a process called kick and sweep methodology along the shores of lakes. And even though it may look like a little bit easy, but it's quite cold, I can tell you, because this is towards the end of October. And the reasons we sample in October and November rather than in the summer is because this is when the so-called invertebrates are taking rest or whatever before the winter. They become kind of docile there, and then during spring they become larger creatures, and then the insects will of course take flight in the summer. So I hope you will all enjoy this, and this is what the general public can do to help, and even volunteers can do this work as well, except they won't be able to analyze these things to a higher level of precision as Angie and Monica can. All right, fellows, so we go again. The lake you're seeing now is Russell Lake. It's only a small lake right in the so-called urban area of Dartmouth. It's right next to the Sobeys cinemas, off Eisner Drive in Portland Estates. This lake had a very long history of pollution going back to the 70s because of various land uses like a pig farm and some chemical dumps and a trailer park discharging sewage with very poor treatment. So in the early 70s to mid 70s, the Department of Environment put up an advisory saying that people should not swim here because they located what's called as toxic algae. But since the late 70s, the trailer park was connected to the main sewer in Dartmouth, which of course leads to the harbor, and then the toxic waste dumps were closed. I'm not sure if they were cleaned up, but they were closed, and the pig farm closed as well. So over the years, let me see how many years is that. That's 20 years. As you know, in Nova Scotia, the lakes flush a lot because we have about 1.4 meters of rain every year on an average year. So I'm assuming here that all these 20 years of rainfall and runoff, which comes from land, mostly from land, the groundwater is just balanced. The amount of groundwater goes into the lake is also replaced with water, so the total resultant groundwater contribution is close to zero. So it's mostly surface runoff, and both the two young biologists working on this project now, Angie and Monica, told me that the kind of macroinvertebrates they found are not indicators of eutrophic lake, and I'm assuming here, based on our own sampling in 91-92, the lake is not as bad as it was in the 70s. So we can all be thankful of that. And another thing to do, all these lands are slated for urban development in the next 10 to 20 years. So we have to make sure that people don't use fertilizers, keep away from pesticides. Siltation should be kept to a minimum. It's difficult to eliminate all of it. So you'll be soon listening to Monica Gartner and Angela Jarvis soon. Okay, people, this is you're now watching a crackerjack biologist by the name of Angela Jarvis, and she's going to give a little bit of intro on this project. So, Angie, why are you here in the water in October, end of October?
Because I'm foolish. Because I wanted a job in biology, and this is what I found. And right now we're looking for macroinvertebrates that live in the lakes, and unfortunately October and early spring is the only time that we can find them. In the summer, as you know, we have mosquitoes and all those nice little pests that like to bug us in the summertime. And in the fall and spring, the adults lay the eggs in the water, and we're looking for the larvae and mollusks and other things that Shalom has mentioned to you before. So, as I said, unfortunately, this is the only time that we can find them.
Okay, now you're meeting the other crackerjack biologist, the other half of the team. Her name is Monica Gartner, and she came here to Nova Scotia all the way from Kitchener, and so we should be very pleased with that. So, Monica, why did you come here all the way, and why are you in the water, in such a cold water?
Okay, I also came because I wanted a job in biology. It was very important for me to get a job in my field. I studied at the University of Guelph in Guelph, Ontario, and I came out here because I was given the opportunity to do a study on the macroinvertebrates of the lakes here in Nova Scotia. We're planning on doing 4 or 5 studies on different lakes, and we're hoping to find good quality water. And how we determine that is first by doing some chemical studies of the water. We take water samples and we have them analyzed, and then what we're doing, the main part, is collecting the macroinvertebrates, which we were speaking of before. What we do is we go out and we kick the bottom of the substrate and we sweep it up with a net, and then what we have to do is sort it out and look for the bugs, and hopefully the bugs that we find will be good water quality indicating bugs instead of the bad ones, and that's pretty much what we do.
Okay, Monica, what are you doing right now there?
Right now what we're doing is measuring the dissolved oxygen in the water. We all need oxygen to breathe, and so do the bugs, so we have to know how much oxygen is in the water. Often when lakes go eutrophic, the oxygen decreases, and this causes a shift in the communities of bugs that we find. It's one of the indicators. When the bugs can live in less oxygenated water, those bugs are the ones that we don't want to find, because that would indicate that we have poor water quality. So right now I'm just getting the reading here. It just takes a couple of minutes.
Okay, Monica, now what are you going to do this phase of the operation now?
Right now we're going to do what's called the kick and sweep. That's where one person is walking along the bottom and picking up whatever they can, and the person has to go behind them with a net. This has a very fine mesh in it, and scoop up whatever is being kicked. We're going to do this for 5 minutes, and we'll let you know when we're done.
Ready to start our kick and sweep. Moni's behind me with the net. The net's almost on the ground, and I'm going to start doing the kicking. We need to be at a meter's depth here, so we are a little bit of distance from the shore. And as Moni's already told you, it's going to take us 5 minutes to do this. Ready?
Yep.
Careful, I just kick up some rocks.
We just have to be real careful when we're walking. There's uneven ground on the bottom here, lots of rocks that are slippery, and we just have to be careful while we're walking.
Now that we're really close to shore, it's hard for Moni to get the net up here, so what I normally do before the jets go by, normally do is turn around and kick towards the net just so we make sure we get everything. Careful.
So Angie, you're kicking for 5 minutes, right?
Yes. We have a minute and 12 seconds to go. Is to move all the larger rocks that are at my feet that you can't see, so Moni can scoop up the macroinvertebrates with the net.
Well, it's just, I'm sure people will wonder too, Angie, have you come across any crocodiles?
No, not yet. But I'm sure I'll let you know if I ever do.
So I guess 5 minutes are over now.
5 minutes over. And as you can tell, 5 minutes can be very long sometimes, especially when you're kicking in the water.
Yeah, plus cold water and everything.
That's right.
Now, Monica, what is all that gooey stuff in that net?
Okay, this is what we've caught with our net after our 5-minute kick and sweep, and what we have to do is just sort of get it all into the middle of the net and then dump it into the bucket.
Okay, what are you doing now splashing water on the other side of the net?
Okay, what we're trying to do is to get all of that muck that you just saw down into the middle of the net so we can get it into our sampling container. You don't want to pour water directly into this because we will contaminate our sample. What I mean by that is anything that's right here in the water where we are will hit into this, so we'll contribute to what we've caught, and we only actually want to catch all the macroinvertebrates that were in our transect that we just sample. So we don't want to add any extra macroinvertebrates to here, so what we do is we splash water underneath the net to move all of our sample down to the center of the net to make it easier to collect.
By the way, people, Angela Jarvis is actually from Lower Sackville, from Nova Scotia. Now I see you two guys are hunched over something that in the trace. So what are you doing now?
Well, what we're doing now is we've collected our sample, and we have to pick through the sample and get the little bugs out. And what we do is we split it up, and each of us count until we've caught 50, so we have a total of 100 bugs. What we're going to end up doing is we put them in ethanol, which is something that preserves them. We will then take them to the lab and analyze them. That means just we look at them and we decide what they are. And we go down to what they call the order level and then down to the family level. We aren't specialized enough to go all the way down to species, but we do try. And then what we do is analyze them by putting the numbers that we get through different indices to discover whether or not the water quality is indicating it's either good or bad.
So Angie, what's the logic behind the 100 insects?
I'm not totally sure what the logic is. I know that they've done different studies and tests using 100, 200, and 300, and they've determined that 100 macroinvertebrates will give the same indice water quality as what 300 will. So just to make it easier, the protocol calls for 100. You will notice that it will take Mooney and I quite some time even just to find 100 because they are very small. So it's just simpler to try to find 100 than to survey the whole thing.
So it's really suitable for volunteers then?
Yes, it's very suitable for volunteers.
Okay, people, you're now seeing one of the macroinvertebrates in Monica's hand. What would that be, Monica?
Okay, this one appears to be a mayfly. This is a good quality indicator. This is what we want to find. I don't know if you can see it.
Yes, I can see it.
You see it? Okay. It's got one of the quickest ways to identify a mayfly is by its 3 tails right on the end there. It's got 3 little tails. And like I said, these are the ones that we do want to find, that they indicate that there's good water quality here because they require well oxygenated water and a good water quality in general in order to survive here. So I'm just going to let Angie put that into our...
Real mayfly was it? But that's the nymphal stage of mayfly.
Well, that's right. The mayfly is going to come out in the spring then. It's going to take flight from the water. What we're looking at right now is only just the larval stages of all of the flies.
All right, Angie, I see some creature moving in your hands, Angie.
Yeah, this is a planarian, or a flatworm, and again, this is something else that we're looking for. They do a lot of oxygen as well, so this is something else that we're looking for, and we've actually been finding a lot of them here at Russell Lake. This is mostly what our samples have been consisting of. So these little guys.
So the lake is not as bad as it was in the 70s then?
No, from the 2 sites that we've done so far, we're getting very good macroinvertebrates, so we're finding that it's a very good water quality, just what we wanted to find.
Okay. Now, Angie, what's in your hand now? I see a creature really moving there.
This is a mayfly, and if you can get a look there at his tail, you can see that he's got 3 little tails, and that's the best indication of what this is. We can see it better underneath the dissecting microscope, but unfortunately we don't have one of those out in the field. This is another good indication of good water quality, so this is also what we want to find.
What's the equipment you're using, Monica?
These are our little pair of tweezers that we use to pick up some of the larger bugs. That's another mayfly. Stick them in the ethanol. And this is just a little pipette. It's sort of like an eyedropper. You just squeeze it and you can suck up little insects. Here's another planarian, and I can just suck them right up with this and drop them into our preservative.
Hi, I'm Angela Jervis. I'm a research biologist for the Soil and Water Conservation Society of Metro Halifax. I graduated in 1996 from Mount St. Vincent University and with my Bachelor of Science degree major in biology.
Hi, my name is Monica Gertner, and I studied at the University of Guelph in Guelph, Ontario. We did a field portion for our project earlier this year, and we showed you that on our kick and sweep, and we just wanted to let you know some of the bugs that we caught during our field portion. These are just some of the examples of organisms that we caught when we were doing our kick and sweep. One of the larger organisms that we caught was from the Odonata order, which are dragonflies and damselflies. This is a dragonfly from the order Odonata. One of the most obvious features for the dragonfly is an extended mouth part, and what it does is scoop up its prey with this. You can see it, it's called a mask, and what it does is scoop up the prey and sweep it towards its mouth. The other sub-order is the damselfly, and you can see the difference. The dragonfly is much bigger than the damselfly. Damselfly is slender, and it has 3 long caudal tails, whereas the dragonfly has just the prongs at the back. You can see the difference. The damselfly also has a similar mask that it uses for getting its prey towards its mouth. Another order that we found is the order Megaloptera. This one is easily recognized by the long filaments on the side of the abdomen. There's 2 different families that we found. The first one has a long, slender tail, and this is the family Corydalidae. We also have one with 2 short anal prolegs at the end of the abdomen, and that is the family Cyalidae. Another common insect that we found was from the order Ephemeroptera. These are the mayflies. This is an example. They are very easy to identify with their 3 long tails. They also have the 6 legs attached to the thorax and gills along the abdomen. This is an example of the family Heptagenidae. Another common order that we found was the Plecoptera, and these are the stoneflies. They're also easily identified by their 2 tails instead of 3 for the mayflies. They also have the 6 legs, and they are missing the gills from the abdomen, but they have very long antennae, which also helps to identify them. Another order we found are the order Trichoptera. These are known as caddisflies, and they will actually be sometimes hard to find because they hide inside of their little houses. They construct houses out of various materials such as grains of sand as well as vegetation. The Trichoptera, when you get them actually out of their house, you can see their 6 legs, as well as they do have 2 anal prolegs at the end of their abdomen. One of the smaller things that we found was a crustacean. It's an amphipod. You can see it has numerous legs on its thorax as well as swimmers on its abdomen. These are much smaller, and we'll show you some real ones as well that we caught. Another organism that we found is from the order Brachiopoda. They're also commonly known as Cladocera. They are very small as well, and they have very large second antennae that you can see, and as well as a tail spine that you can see at the end there. They're also, you need a microscope in order to see these organisms better.
We just thought you'd be interested in seeing some of our live, well, they're preserved now, but some of the actual specimens that we collected out in the field. These again are the dragonflies and damselfly that Monica was discussing earlier, and we just wanted to show you the mouth parts that they have This is the hinged mask that Monica was talking about, and you can see it a little bit better on this specimen. Unfortunately, the damselfly is smaller, and you can't really see its mouth part, but it has the similar masked hinge that the dragonflies have. These, again, are some more specimens that we collected from the field. These are the Ephemeroptera that we showed you before, the picture. We just wanted to show you some of the different sizes that you can get. Unfortunately, not all of the Ephemeroptera come with the largest specimen that you see here. This one is Heptagenidae, and you'll notice that they do come in quite a size range. Unfortunately, we didn't find too many of the larger types. It makes them easier to identify. You can clearly see on this one the 3 tails that we were talking about, and again, the 6 legs. The 2 Megalopteras that we were talking about before, you can clearly see the filaments on both of them and the differences in the tails that we were pointing out, but the major identification for the Megaloptera are the filaments that you see on the side. This is the Trichoptera that we were talking about. Unfortunately, you can't see it as clearly as what the book indicated. They are a little bit smaller in size, but basically what we want to show you in here is this is actually one of their houses, and we've just pulled this little guy out, and as Monica was saying to you before, they build their houses out of anything that they can find. This one is built out of small twigs and things like that. The Trichoptera that we were discussing before, and again, the most distinguishing feature of this one is the 2 tails. They have 2 tails instead of the 3 tails that you see on the Ephemeroptera. So in the field, this is the easiest way to tell them apart. These are 2 more specimens that you haven't seen yet before. Well, at least not from us. You might have seen them if you've been walking along the shoreline or something. One of them you might really not want to find. It is your common leech, and it's the crescent-shaped specimen that you see there. Unfortunately, you can't see its most distinguishing feature. It's the 2 suckers that are on either end, the oral one and the posterior one, but most people know what a leech is. The other one is a roundworm, or an Alicoceta, and we found quite a few specimens of these. They're really easy to see in the field once we scoop them up in our net and place them into a little bit of water. You can tell the movement quite clearly from them. They like to squirm up and elongate once you have them in a bucket. So they're really easy to identify in the field as well. 2 specimens that we collected. They're from the order Gastropoda, and the smaller one on your left, my left, would be a bivalve, and that would be what you would find in the ocean, a clam. And the one on the left, which you can barely see, is the common snail. And we found very few of these, but we did find a couple, and we just wanted to show you a sample of them.